Okay, so this is the second part for the glycolysis video. In this video, I am going to discuss about the substrate level phosphorylation. Now, what is a substrate level phosphorylation? Related to glycolysis, there are two steps where substrate level phosphorylation takes place. Now, what is the meaning of the word substrate level phosphorylation? This means that when, suppose in this reaction you can see, when from 1,3 bis phosphoglyceric acid 3-phosphoglyceric acid is forming, then in this particular one, this is 1, this is 2, this is 3. So 1,3 bis phosphoglyceric acid. So in this molecule there are two phosphate groups, this number 1, number 2. Now this is another compound 3-phosphoglyceric acid, where only in the 3 position of carbon this phosphate group is present. So this one is not there. So ADP, adenosine diphosphate to adenosine triphosphate, that means one more extra phosphate group. This is tri and this is di. So this phosphate is attaching to this thing and it is forming ATP. This is the cofactor and this is the enzyme that is required, phosphoglyceric acid kinase. I have already discussed about this. Now as you can see over here, this is the substrate and this is the product. This is the starting material, that is the substrate. So substrate level phosphorylation is a type of reaction in which the substrate donates both the phosphate to form both the phosphate and the high energy. As you can see, this high energy is also provided. So when the substrate will itself donate both the energy and phosphate to form a high energy bond, high energy molecule in the absence of oxygen, then this particular type of reaction is known as substrate level phosphorylation. One more example of substrate level phosphorylation in glycolysis is from phosphoenol pyruvic acid to pyruvic acid. Here also you can see that this is phosphoenol pyruvic acid. From here to pyruvic acid, this phosphate particular group, this high energy ATP bond, both are given to this thing to form ADP. This is the enzyme, this is the cofactor, this is the coenzyme to form this thing. This is pyruvic acid. So these are examples of substrate level phosphorylation. Next, we will be talking about oxidative phosphorylation. Now, what is oxidative phosphorylation? Oxidative phosphorylation is a type of reaction in which, see, there is also a requirement of, there is also formation of a high energy bond over here. But phosphate is also there, but here we have to add PI, meaning inorganic phosphate. Here also we know NAD plus is used, which is going to convert to NADH plus H plus. And the enzyme used over here is glyceraldehyde. Glyceraldehyde 3-phosphate dehydrogenase. So here we require oxygen to form a high energy bond, right? So this is the basic difference, and oxidative phosphorylation in glycolysis is from glyceraldehyde 3-phosphate to 1,3 bis phosphoglyceric acid. Simple as that. Next, we will be talking about inhibitors of glycolysis. That means the particular type of low ligand or any molecules that will inhibit the catalysis of enzyme. That means it will not allow the enzyme to work properly. That are known as inhibitors. What are the 3 inhibitors? That is iodoacetate or iodoacetamide, and it will inhibit glyceraldehyde 3-phosphate dehydrogenase. This is number 1. Bromohydroxyacetone phosphate, which will inhibit phosphotriose isomerase, and excessive fluorides will inhibit enolase. This is Mg2 plus dependent enzyme. Next is rate limiting steps of glycolysis. That means the steps that determine the rate of a particular metabolic pathway. So from glucose to glucose 6-phosphate by glucokinase or hexokinase. This is one rate limiting step. Fructose 6-phosphate to fructose 1,6 bisphosphate by phosphofructokinase. This is another rate limiting step. And phosphoenol pyruvic acid to pyruvic acid by pyruvate kinase. This is another rate limiting step. If you are asked about what are the rate limiting enzyme, then this will be the rate limiting enzymes. And you have to remember that rate limiting steps, if I say, should have to be irreversible. So these are only the 3 irreversible steps of glycolysis, rate limiting steps of glycolysis, and these are the rate limiting enzymes. So here we end about this glycolysis part. If you have any doubt in the comment section, you are free to ask. Thank you.